I'm delighted to be here. I'm Corby Comer from The Atlantic to be with Carla Schatz, who is a professor of biology and neurobiology and director of Biotech at Stanford University, previously chairwoman of neurobiology at the Harvard Medical School. David Agus, who many of you have heard multiple times here, professor of medicine and engineering at University of Southern California and leads the Westside Cancer Center and Center for Applied Molecular Medicine, author of the best-selling End of Illness and A Short Guide to a Long Life, both of them New York Times bestsellers. And we have Dennis Charney, a world-renowned researcher and transformation leader who's leading the Icahn School of Medicine at Mount Sinai to unparalleled growth and high national rankings. When he was at Yale, he worked on anxiety in a way that began this entire session when I did an hour discussion with my beloved colleague Scott Stossel, author of a superb book called The Age of Anxiety, about the history of anxiety and psychopharmacology in general, which heavily quotes Dr. Charney. So I'm delighted to have them all, but today what we're going to be talking about are the big gaps in biotech. What's preventing it from going forward? Has it kind of come to, as there's an end of illness, or are we at the end of biotech? What's happening with funds and regulation? I thought, though, that what we could start is to have Carla, I'd have to call you all doctors, so let's just do first names, is have Carla talk about the beginning of the pipeline. We all think we have an idea of the drug pipeline and where it starts, but how do you define where it starts and who supports the various stages?
Oh, sure, I'm happy to do it, and I'm happy to see everybody today. So, first of all, let me just say I think there's sort of good news here, too. Mm-hmm. And, you know, our country has invested over probably now 50 years in biomedical research, and in particular in the beginning of the pipeline. And from that investment, we now have sequenced the human genome, and we know something about really the building blocks of life. And that's the beginning of the pipeline. That's sort of the fundamental knowledge about life systems, you know, human biology, that has helped fuel recent advances. And what, you know, we talk about is personalized medicine. Mm-hmm. So that's really at the beginning. And that, I think the beginning of the pipeline is also work in the fundamental nature of cell division, of cancer, of how our brains work. So in other words, you know, when you go to your doctor and you ask her for a pill to cure something or to take for treating something, often you imagine your doctor might be the person who's involved in inventing and in the cure.
I'll just say, if we go to Stanford in your department, I think we're probably imagining right.
My department. Well, which one? Neurobiology?
See, which one?
Yeah.
But in any case,
well,
it's usually the
exception. With David Agus, I think that's true. It's really the exception rather than the rule, because often these cures are coming from fundamental discoveries that have been made in how things work. I mean, shall I tell a story about an example of a cure, a treatment that's...
And I'm going to say no when you've just asked, should I tell a story?
No, go ahead.
No, please do.
Say no.
Please do.
No, you know, we now, Alzheimer's is a very, obviously a huge challenge now for our population. And in fact, at this point, we know some of the genes that cause Alzheimer's, and that kind of knowledge has become the basis for the development of new drugs. And time will tell whether these drugs work, but they are actually the fundamental basis. And one of the genes that was discovered that causes early onset Alzheimer's disease is called presenilin, and it was discovered by doing human genetics and studying families and trying to see what kinds of genes they carry that might be mutated or altered that would cause the disease. And nobody really understood precisely what presenilin did. And it was hard to then develop a kind of a therapeutic approach without knowing the function. This was all happening in Washington, in New York, in Boston. On the other side of the country at UC Berkeley, a scientist was studying yeast. What does yeast have to do with Alzheimer's? Long story short, one of the genes that was involved in the way yeast reproduce and bud turned out to be identical to presenilin. And the scientists there, doing fundamental work, knew everything there was to know about this presenilin gene. And because of that, new therapies are under development at this time. So I think it's that kind of wonderful collision between science and medicine that sort of illustrates how the pipeline gets started. There's a disease and there's a discovery, and often they happen in different places, and sometimes they happen in the same place.
And because this will be an underlying theme of this hour, who funded those two groups?
Oh, actually very different sources. Mm-hmm. So I don't know because I wasn't involved in those studies, but I'm assuming that the National Institutes of Health funded a lot of the work on the human studies, on the genetics, the human genetics, and on the discovery of what was called presenilin. On the other hand, the Howard Hughes Medical Institute funded a lot of the work on the yeast story. And why did they fund it? Because it was fundamental, fabulous scientific research. By the way, I don't know if I said this, but this yeast geneticist won a Nobel Prize for his work, in fact. It's really amazing. His name is Randy Schekman. And the Howard Hughes Medical Institute did recognize that it's worth investing in what might seem at the moment to be just great science farthest from translation.
Is that not what's called basic science?
It is, absolutely. That's what's called basic science.
So who is funding it is the question. Dennis, since I'm going to count on David to throw flames. What me? Dennis, can you lead us through some kind of drug discovery and development and where basic science support came from and who came in to really bring it to the next step?
So the data that we all are in our field are familiar with is that it costs roughly $1 billion to take a drug to market with the classical approach, which obviously is not fiscally feasible. But those have been the numbers. And that's in part due to the fact that we haven't had good enough targets for drug development. They're not predictive enough. There's not enough collaboration between the NIH, which funds most of the basic science, and industry, which is risk averse. So that leads to the number of $1 billion because there's so many failures, going from having a molecule that might relate to a target, then you develop a lead molecule, which is closer to being a drug, and then you develop a drug that might get into phase 1, which is the early phase development, looking for safety in humans. And then it gets into phase 2, where you're testing the drug in a clinical population, and then large-scale studies are done in phase 3. So it starts from a basic science lab, identifying a target, and then gradually into human populations. Along that pathway, there are failures, and because we don't have good predictors, and that leads to the enormous cost in getting a drug to market. However, as Carla is alluding to, that the advances in the human genome and advances in basic science and advances in cancer in particular, where therapy can become more personalized based on genetic abnormalities in tumors, that we can develop new models for drug development. Mm-hmm. That's exciting to all of us in the field. However, there are not the funds. So the NIH budget has, in real dollars, gone down. It's never been harder to get a grant. So 90% of all meritorious grant applications do not get funded by the NIH.
Can I just ask about that? Do you mean 90% of decently filled out applications, or are you saying, you know, there's many more, but 90% of the good ones don't get funded?
So here's one way of defining it. When you submit a grant, it has to reach a certain bar to even get a score. And if you get a score, it's considered to have merit.
Right.
And then you get a score, and then there's a question of whether it's funding. 90% of all the grants that get a score are not funded. So we're losing a generation of young people because they're not able to get grants.
But can you tell us about, you know, bring us to the glory days when you actually were able to develop a drug and you were able to get funding? Because, you know, she's ahead of you with the mouse yeast story. So do you have any actual stories of drug development that worked?
Well, in my field, which is neuroscience and psychiatry, most of the glory days were serendipity, meaning that, you know, we discovered drugs for depression and psychosis and anxiety, not having a clear hypothesis, but astute clinical observers noticed the clinical effect, and those drugs were then developed and worked to a degree. The challenge now for neuroscience, given the complexity of the brain, is to have a priori hypotheses that then lead, you know, to an effective treatment. And we've got lots of ideas now. I think the main problem is funding, between industry backing off in neuroscience and the NIH budget being so difficult.
Are you watching that happen at your hospital, in your department?
Yes, it's, you know, like every major medical school, the challenge of getting grants is enormous. And the average, here's another fact that's, I'm generally an optimistic person, but
Just remember that, Evan.
the
I
average
think we all are.
age of getting your first independent investigator grant from NIH is 42. Now, for most of us, that's mid-career, right? That's unacceptable. That means when you're in your late 20s and your 30s, you're not getting funded because it's so difficult to get funded, and we're losing a generation of investigators because of that. And that's true at Mount Sinai, Stanford, USC, you know, all the top schools, but it's even tougher for the schools that, you know, generally are not at the top.
David, thank you. David, what's missing in this? What are things that the government could do, or who should be putting money into this, and what's stopping it from going in?
There are a lot of ways to answer the question, but you know, we got screwed in the late 1990s by something called the internet. So the internet screwed biotechnology, because all of a sudden, if you're an investor in the capital markets, you get a return in 2-3 years, and it's a big return. You want a return in biotech, it's 12 to 14 years to profitability. Are you going to put your money away for over a decade and hopefully get a 10% return a year? So all the capital moved another direction. All the great venture capitalists got out of biotech and started to go into technology and internet and the lifestyle devices, you name it, they got away from this field. And so the NIH was still there, NIH funding staying relatively constant, yet obviously inflation is happening, more investigators are happening, so we're all being crunched for dollars. The NIH has two budgets. It's got a budget where they give people like all of us money to do research, and that's based on grading scores. And they've got a budget of what they do internally. And in a sense, they compete with each other. Many of those internal projects are science projects, basic science projects. And historically, they were great, right? During the Vietnam War, they were called the Yellow Berets. They didn't want to go fight in the war, so the best and the brightest in the country went to the NIH to do research. Right now, though, there's a call to arms. When this is happening in our country, we need to start to do things. Wouldn't it be neat if the NIH used its internal intramural budget to start to develop the resources so we could develop drugs better, to do the pharmacokinetics for us, to do the quality metrics, the building of drugs or the producing of drugs for us so that they can enable us to do it as an academic institution?
Let me back you up because that sounds so good. Who's bearing those costs now? Who did bear them, and have those people backed away?
So it was the capital markets, right? You had a great idea, you developed a good molecule, the venture capitalists gave you money, you went public very quickly in the 90s, you were able to raise $20, $30, $40 million and put together a good shot at a phase 1 clinical trial. Mm-hmm. If the initial trials worked, big pharma would come in and they would give you the money for that next stage in development, and it really was a good pipeline. And that pipeline's starting to dry up. Biotech over the last decade has started to die. There aren't great new companies. The Genentechs in the world, and by the way, Genentech is gone. It got acquired because it failed. because again, the capital requirements. There aren't new great biotech companies, a couple exceptions here and there, but certainly not dramatic ones. And that's causing a major problem for all of us. There's that divide, great discovery, but discovery means nothing if you can't get it to benefit patients. And we have to push hard. Big pharma is awesome, but they want things that'll be $2 billion in sales by year 2 or 3. Most of the diseases we're talking about, initially they're going to be very small sales. So they say, listen, I'm not going to worry about a $200 million drug. It ain't worth it.
But there are things we can do. And one is, in many fields, Me Too drugs are developed. So in psychiatry, with the antidepressants, there are 7, 8, 9 different antidepressants that have the same mechanism of action and don't work any better than each other, but they're each $1 billion drugs. So the companies are incentivized not to innovate, not to take a risk, to have to spend the $1 billion, to develop a Me Too drug that gets approved by the FDA and still makes money. There should be incentives coming out of the FDA, and they're, I think, working on this.
You don't mean financial? FDA has no money, right? So what kind of incentives?
Reviews, life of the patent, how long you have patent life, fast tracking of reviews. So all that can be done to incentivize industry to innovate as opposed to developing Me Too drugs that don't advance the care of the patient.
What if you allow biotech companies, and they have losses for the first 10, 12 years, to actually sell those losses to another company so they can get the tax credit for them? So instead of delaying their tax credit on their losses till they're profitable, they can actually use that to generate more capital to develop these drugs. Relatively simple thing to do.
It's a great idea. I mean, I think this issue of risk is a really big topic that we could spend a lot of time on. But this idea is wonderful because many of the startup companies that were funded in the past didn't have enough time, really did work on and develop fabulous new intellectual property, actually beginning to move compounds toward making drugs, even having some of them work in the animal models. But because the clock was ticking and it ran out, the companies actually then were either sold or acquired, and all the work that was done ended up buried and not available. And this is, so allowing these companies to have a longer lifetime would be a huge, it would allow risk to be absorbed in a way, and it would also allow for new discoveries to see the light of day.
When we were talking earlier, you mentioned something about all this lost data, these experiments. The companies go under, it goes into a black hole. Does it really? It remains proprietary until they can find more funding and it can be, revivified like Brigadoon or
Sounds
something?
like a TV show.
Right, exactly. Like it's cryogenic data.
If you do a clinical trial and you put a drug in a patient and you spend 10, 20, $30 million and the drug doesn't work, you don't want to put your data in the public domain. It could help your competitor. This is your child. If it does work, you even more so don't want to put it in the public domain. Yet the heroes are the patients. Patients are taking these drugs and it's their data, not the pharmaceutical company's data. So we really need to look at ownership of data. We need to look at the ability of putting data out to journals. What journal would take a negative study? If you have a study that didn't work, the good journals say, I don't want that. I want the ones that worked. So how do you publish negative results and get it out there?
Or even the idea that the FDA, there are certain ways that you do a clinical trial. And what if in that trial, even though overall the patients didn't, let's say, respond to the drug, maybe there's a subset in that trial that did respond, that would be worth following up on and understanding. Again, it's extremely difficult to explore that kind of information or even make it public.
The NIH is doing something good in this regard, where they have an initiative involving repurposing of drugs that are approved. So there are, or even past safety requirements with the FDA.
Off-script.
Excuse me?
You don't mean off-script kind of use?
No, not
Off-label, yeah.
No, not that, but that's another issue. But that there are lots of molecules that have passed safety so that they could be given to humans that have not shown efficacy for one particular disorder, but might show efficacy in other conditions that have not yet been tested. The NIH has an initiative with pharmaceutical companies to give researchers access to those drugs to test them in other indications. That could fast track the discovery of new treatments.
David, when you were saying earlier, what about being able to sell the risk and kind of amortize it and keep it going forward? Who would put that into place?
There's a small group of people in Washington called U.S. Congress. And so...
No one's heard of them here, have you?
You know, I mean, again, Congress has to get off their butts in this regard. When the FDA was established in the 1950s, genomics didn't exist, proteomics didn't exist, and so all these great new technologies that you alluded to that can help us develop drugs, so Congress didn't give them, the FDA, the authority to regulate them. So all of a sudden, these technologies are here, and they yet have to give the FDA the authority. So it's not clear who regulates it. Well, if you're a smart venture capitalist and you say, listen, I don't know who's going to regulate this, I don't know what the hurdles are going to be, why would I put my money in? It was, in fact, Senator Obama at the time, his first bill was this, the Personalized Medicine Act. And what happens is they just keep attaching a bridge at the end and a tunnel at the end and whatever, and it never goes anywhere. We need to actually take some action here, because who doesn't benefit are the people with disease, which is a good fraction of our country as they get older.
So a somewhat contentious term that is, infinitely popular here is personalized medicine, and it came up this morning in our discussion, and you've come out with it now. And one of the reasons you were mentioning it was, if you can't develop a drug that's going to work on a large population, and you can find a subpopulation on which it works, then you can develop that. So is that something that can be profitably and more quickly, let's say less slowly, pursued by researchers, by pharma?
Yeah, I mean, there's a couple definitions of personalized medicine that relate to drug development and the use of drugs. The lowest hanging fruit is that our field has identified genes that relate to side effects with drugs. And the FDA has begun to recognize that. So with the electronic medical record, we can get information to the doctor in their exam room based on genomic testing saying, don't give this drug because it might cause a side effect that's dangerous. On the other hand, in areas like cancer, diabetes, and some other areas, there are, like breast cancer, there are genes that predict response in a subset of patients. That's helpful now, and it can be helpful in drug development if we design the studies ahead of time saying we'll do the genetic testing and we're going to test the drug in this subgroup. Then you get a higher chance of response.
You know, on this stage a couple of years ago, I was in a session with Murray Gell-Mann, one of the great physicists, who discovered the quark and et cetera.
Who is here?
And he looked at me in the eye and he said, my field succeeded. He goes, even myself, that's how I identified the quark, is by looking at outliers. I saw these numbers that didn't make sense. And that outlier, I was able to make some of the fundamental discoveries in physics, and so did the Einsteins of the world. In our field, we discard them. Larry Summers made a great example where you say you could take your card and you will go to any country in the world, you put it into a box, 11 seconds later, it goes to your bank, verifies it's you and their funds, and you get money out. If I said to a patient, I want to use your data and pool it with everybody else with your disease to look at the responders, the non-responders, the outliers, that's a crime in our country. So we need to say, listen, we need to call to arms. We all have to be heroes, and we have to put our data forward for our kids and our grandkids, and hopefully us, because we're going to learn from it. We're going to find those outliers, those people with the side effects, then we can look at their genes, those people with the dramatic responses, and then look for markers, but we're not doing that now.
Yeah, that's something I want you all to, remember if you leave here. There's a point I'll make at the end, which of course you want to remember, but a very big point is data sharing. Data sharing. So Dennis, I think that your hospital group, Mount Sinai, is taking a stand on trying to make data more accessible across departments and across hospitals that I'd like you to tell us about. And Carla, you had some thoughts on this too.
Well, that's true, not only within our healthcare system, which is now one of the largest in the country, that we are sharing across the system, and that helps innovation and come up with new ideas. But our research group, some of which have been at this conference, particularly our genomics group, is totally wedded to open source sharing of data that will then help all of us make innovative new discoveries. Classically, medicine has been conservative, traditional in how we train our doctors. The The curriculum that we have now has been in existence for 100 years. We tend to attract students with a fairly narrow interest and are not getting the engineers and the computer science majors and the math majors that are part of the digital revolution. Mm-hmm. By making changes in who we recruit and how we educate them and being more open source in how we share our data, that will lead to more entrepreneurship and innovation.
So we really have to train our scientists and our clinicians to collaborate with each other and to know how to do team science. And actually the X in BioX, which is the program that I direct at Stanford, is really the intersection of disciplines. So the idea is to get out from under the lamppost and really look at how complex the problem is you're working on, and then collaborate. So if you're a neuroscientist, you might want to work with an electrical engineer to make a really cool machine that you could implant in the brain of a paralyzed person, so that person, just by thinking, could actually move a cursor or move a robotic arm. These things are actually happening. And I think we really...
We saw it happen at the opening session.
Oh, you saw that?
We saw somebody moving an actual device, a robotic device, with his eyes over the internet. It was the most moving thing.
Yeah, it's quite unbelievable. And it's this kind of idea of team-based science, and again, the mixing of the physicists and the chemists and the engineers and the biologists and the clinicians, so everybody is in the team, is probably one of the only ways we're going to be able to tackle some of these complex problems going forward.
I agree, but it's a culture change. And I wrote a piece once, pushing for this, and I said, because right now only the first author and the last author on a scientific paper or a medical paper get credit. Nobody in the middle gets credit and they can't be promoted. So if you have team science with 5 people, how are they all going to get promoted and have a career?
Well,
so I said, let's do it alphabetical. Since I'm Aegis, reverse alphabetical. And you put next to the person what they did. I got letters from 7 university presidents saying, David, this is inappropriate. This is not what academics are about. We can't do this. We have no criteria. And they were offended by it.
Well, we should do this together.
We're doing that.
That's great.
At Sinai, it's not first or last. It's you say what you have done, and that's what goes to the appointments and promotions.
Yeah,
great.
so this is actually what exactly at Stanford, and it actually does require a fair amount of work to constitute a committee that can evaluate an assistant professor for promotion. But that's exactly what has to happen. And obviously we all have to do it, otherwise it's not going to work.
I was trying to get someone promoted. There's this remarkable woman who had worked on 10 different projects and made sure they got done, and we're putting up for promotion. And the provost said, you see that elevator? This building would not run without the elevator. Without that elevator, nobody could do science, nobody could do medicine, there'd be no discoveries. And I'm not putting that elevator up for promotion.
What did he mean? How is he comparing an assistant professor and an elevator?
Because they didn't have the first author papers. They were part of a team that made some accomplishments, and they were a key part of it.
What an enlightened attitude.
I know.
Something that you've all pointed to is the idea that in the most basic and exciting research, and in biotech, and for it to go forward, outliers have to have a chance to be heard. And where are you in the mainstream going to see outliers? And in an age before the internet, when venture capitalists were more excited about biotech than they are now, not demanding such quick returns, was there a chance for outliers to be seen? The crazy garage biochemist. I mean, was there a system that embraced new ideas, as you're all implying needs to be done, and built into the system?
I want to give an example. I think this is all about risk again, but it's about spinal cord injury. So I was on the advisory board of a company that was acquired. So it didn't quite totally fail, but in David's viewpoint, it did fail because it ran out of money. This company was founded. No, we're not going to name this company. This company was founded to work on cures for spinal cord injury, based on major discoveries that had been made in the fly, believe it or not, because people could study the same genes in the fly that were important for the growth and regeneration of nerve connections in humans and in mice. And so there was a huge amount of really wonderful fundamental basic science that was used to form the company. And the company failed because and it was outlier in the sense that many of these discoveries were not mainstream at all at the time when the company was formed. In fact, that's what made it attractive, I think, for people to invest in the company, because it was the first. But after a few years, it was clear that the problem was going to be very hard to solve. And so the company then was sold. And that part of the company disappeared, and all the intellectual property disappeared. So
when you mean disappeared, you mean it went into a vault?
Yes, it is in a vault, actually. The developments in the company are in the vault. Luckily, neuroscientists have continued to work on the problem. And thank God, all the molecules and all the fundamental discoveries were published and are publicly available. So people are still working on the problem. But the difference now is that the same venture capital companies, many of them that were willing to take the risk in the old days, don't want to take a risk now. And so there's this, what we call the valley of death, which is a valley where these discoveries die before they can go into the clinical translation. So they're made, they're really cool, they get published in great journals, and then nobody will pick them up to allow them to go forward to be explored, and even for the first compounds to be made that might later be turned into drugs.
So I think what happened also is that when there was a lot more venture capital money available, that the investors weren't, I think, fully cognizant of the risk and how long at that point it was going to take to develop a drug and get it to market. The numbers, like I mentioned before, $1 billion, 15 years of research to get it to market. But that is now changing, and that's kind of the opportunity on one hand, and what's unfortunate is that the science is better, the genetics is changing our ability to predict, but now the investors are more risk averse when they probably don't have to be at this point because our methods are better.
It's also our fault, though. It really is also our fault. In the 1960s, you had a group get into a room and say, if we want to exchange dollars across the world, this is how we have to do it. And they set the data exchange standards for dollars that allowed credit cards and other things to explode. Happened in the 90s, a group of 12 people got in a room and set up the standards for the internet. We don't have data standards. So you call it a broken leg, I call it a fractured leg. So what each of us do, we have our own databases, our own clinical trials, yet we can't put it into one common one and learn. We can't learn from each other because we're all using different terms. So we need to, as a field, stand up and actually start to make a statement. Establish these data standards, establish standards on collection of data, deal with privacy. While it's all good to put genomics information out there, right now it's not clear it's legal. If I put your genes out there, there's something called the Health Insurance Portability Act, where privacy-wise, no matter how you do it, I can identify it's you.
How?
Well, I can look back at the DNA, and there's a beautiful paper published in the National Academy of Sciences showing how from just your DNA sequences, I can figure out that it's actually you.
Okay, I know it's bad enough that when I go onto sites I shouldn't be looking at, my IP address will always get back to me. But you're saying there's some way always of tracing even a random bit of my DNA?
Yes, so we can take...
But how again, even if no one has ever theoretically attached that to my name?
Well, we can look into databases and figure out things like your race, your age, and others that correlate with certain parts of DNA. And then in the end, we can figure out it's you. And there's a beautiful paper that went through the decoding, if you will, to show that that was possible in almost all cases. So we need to figure out better ways of doing this.
So doesn't this plunge you into despair because it means there will never be the free sharing?
Who cares if somebody knows if you're the DNA? That's what we have to change, is the attitude.
My life insurance company, of course.
Well, life insurance does not grade based on DNA right now. And there's also something called GINA, the Genetic Non-Discrimination Act,
right,
that President Bush, and one of the few good things he did was actually get GINA passed so that you cannot discriminate based on DNA. So we have to change the culture. And to say, listen, there is risk here, no question about it, but we got to stand up to it. And we have to say we need to be part of the solution.
Can I just add one other element here?
Dennis.
And we haven't mentioned philanthropy. And when you look in total, the challenges in biomedical discovery, with the NIH budget being tight, investors being more risk averse, I think the case can be made that philanthropy is never more important, because that allows us to take risks, make unexpected discoveries, look at the outliers who respond and interrogate their biology. And that has already made a big difference in several major areas like the Broad Institute at MIT and Harvard. A lot of the philanthropy that we get at Mount Sinai is critical to the engine of discovery that we have. The same is true, I'm sure, at your university. So that is a critical element right now, probably more than ever before.
I mean, I think we're all incredibly grateful for philanthropy. I know I am in my own lab. There are discoveries we never would have made if it hadn't been for a really, a visionary person who gave us enough money to do the first sets of experiments. But one interesting thing about philanthropy is that, rightfully so, philanthropists also care about very specific diseases, for example. And I think it would be fabulous to even broaden the idea of philanthropy and remind people that discovery and cures start even before disease. And so what we actually need are more funds for really good science that don't necessarily have to be targeted. It's the yeast story all over again.
That's a much tougher sell for
yeah,
philanthropists
absolutely.
who had a family member die of a certain disease, and this is their dedication. It just makes it harder.
Well, there's a group at MIT that published an amazing paper in a journal called Nature. And what they looked at is a new way in all these Greek letters to fund biomedical research, where if they start to pool risk, like they did in the housing market, although I guess that result wasn't that positive. But the hope is they could do that here. And when he calculates it out, it almost guarantees an 11% return in this beautiful paper where he takes different stages in disease, different diseases, different goals, different sizes of disease, and lumps them together into one large fund. So clever ways of putting the capital together are needed.
That certainly sounds good, and everyone should be, convinced of it and just take it on faith because Nature published it. Do we have any questions? We have a mic, and we have 2 hands up at this table. Orange.
Carla, Judy and Bob here, you've written to us to say thank you for the money we gave you for basic science. And
Did you mean it?
Yes,
and we
absolutely.
keep trying to meet you, but we haven't had the chance to.
Thank
But
you.
the reason that we didn't give you a lot, but we gave you what we could, and we gave it to you not only because we believe that the basic science is the building block that leads to all the rest of the stuff that you're all talking about. And if we don't do the basic science, it isn't going to happen. And also because we wanted to use this as a teaching opportunity for our grandchildren who are young teenagers. So the BioX lab is stewarding not only Bob and me, but also our granddaughters. So they're getting research updates about what that money is being used for. And what we said to them when we gave it to them as a holiday gift, we gave them this donation, we said, you're living through an amazing revolution. We've lived through the Industrial Revolution, the Information Revolution, and now we're in the Biomedical Revolution. We're not going to be here to see what happens as a result of this money, but you are, and you're going to see what happens, and you're going to contribute to it in your lifetime.
We're going to put up a short video of just what you said and try to inspire many other philanthropists. You and then.
Yeah, no, I was going to ask you about the CPATH Institute in Tucson. Whether any of you are familiar with it and the work that they're doing, because you're talking about data sharing and biomarkers. And
CPATH is a remarkable group, and just an amazing woman just went from the NIH to help run it, and she's fantastic, Carolyn Compton. And it's the idea that develop a critical path and start to put resources there to make things happen quicker. And so that needs to happen. We need advocates for us, for the industry, for drug development, to help the FDA get the legislation it needs. And they're an avenue to do that. Obviously, their funding is minute compared to what needs to be done, but they're a great institution.
woman right there.
I have a quick comment and a question. I'm part of an investment group that invests in early stage companies, often life sciences. And so one comment, since this hasn't been said, I want to say that we believe, and we think this is right, that many of these companies that may take a very long time to get to an exit will have points of inflection along the way that will allow them to exit, which can make it very productive for investors. And we look for those opportunities, and we think they often exist. But my question is whenever we consider a life science company, we frequently, and probably too early, start evaluating the likelihood that there will be insurance reimbursement for this thing, whatever it is that is being evaluated. I'm just wondering, do you get into that at these very early stages, or is it just not worth spending time on at that early
Listen,
stage?
if there's no insurance reimbursement for it, in general, it's probably not the greatest drug. So insurance is a smart business. If something works, they generally pay for it. And so you can argue on technologies it may be a little different, but certainly on drugs, if it works, they pay.
Yeah. You know, in one area that we haven't discussed, but it relates to your comment and question, is, and I think maybe this was discussed at some other venues here, is that is the development of new applications for delivering clinical care in the home, and also developing new apps that can actually be therapeutic in some way. And insurance companies are very interested in that, in working with those products with the idea that they would improve wellness and reduce costs.
All right, a question right there.
Thanks for your comment on the investment. So I work with life science companies, usually startups, young companies, on their business plan, their business strategy, finding money. And I would just offer this comment. We're in a pretty incredible country right now. My Japanese clients want to enter the market here. My Korean clients want to enter the market here. My Spanish clients want to enter the market here. Life sciences is big. It's growing. It sees good returns from venture capital. Yes, in Silicon Valley, it's difficult to get dollars because you're competing for IT. In Cambridge, Mass, it's challenging because there's so many great companies doing really great things. And there is what you call this valley of death, but there are a lot of new alternative investors coming to invest in that space, foundations, nonprofits like Leukemia Lymphoma Society, philanthropy. There's a whole long list. So the challenge is really not, is there money? It's helping young entrepreneurs figure out where to source the right fit for what they're trying to do.
And
And the other side of the coin, how to make the match between the young entrepreneurs and your companies and the discoverers, right? The people at maybe universities or in hospitals. Frequently, I've seen. I actually, part of my job is to try to make those sorts of matches. And frequently, what happens is I see that the investors come, or the pharmaceutical company comes, and it's read the neatest, hottest paper in Science magazine, and so it wants to only talk to so and so. Whereas we know that there are so many amazing discoveries that are happening at Stanford. It doesn't matter where, and the matches are also not being made as well as they could be.
You know, the greatest venture capitalists in the 90s were all in biotech, and it was awesome. And they do a lot more than just provide money. They help the entrepreneur, they make you successful. And most of those firms have left the biotech arena.
So they were like their own incubators.
They helped you I'm a scientist. Do I know how to make a company? Do I know how to make things work? They made it so I could be successful.
Yeah, and we're exploring another model, and a couple of other places are too, and that is we're taking some of our endowment and creating a venture fund internally with competition among our scientists to develop new ideas that have the potential for commercialization. And then we're moving that along the development pathway based on our venture funds, but then partnering, looking for partners, in the life science industry. So it's more of a hybrid model, using our own internal venture funds to get things started.
The young man.
Uh-oh.
Right now, there's a lot of new technology coming out to help better creation of genes? And how come that hasn't helped further the speed of creating a drug? Like at UCLA, you can see viruses on cells and how they react with drugs. How come that hasn't sped up the process?
Well, there's a time lag between the kinds of discoveries that we're talking about in identifying genes that relate to cancer, vulnerability, genes that relate to mental illness, and using that information to develop molecules that will lead to new drugs. So we think it's probably coming, and that's why we're a little more optimistic than we were years ago in terms of the pipeline and the cost, but there is a time lag.
But one thing, I love your question. It's one of my favorite questions because my question, to rephrase it, is, we know all these genes, but how come we haven't got cures? Alzheimer's is, again, going back, a really good example. We have a bunch of we know a number of genes that can cause early onset Alzheimer's. Why don't we have any cures? And part of the reason is not just, the discovery process, but the system is complicated. So even if you got the gene, you put it back in the animal, it may not work the way you thought. So the cures don't necessarily work because you don't really understand the complex systems. And if you just think even about cardiovascular disease, we have Lipitor. But we really don't know what are the underlying mechanisms that cause the long-term changes in cardiovascular tone. Why do plaques build up over time? All this stuff we don't actually have the answers to yet.
You're in
So
David's territory.
I am in David's territory. And so there's a lot left for you to do.
Yeah, we need you. If you're going to drive from Aspen to Denver and you're going to take apart a car, look at every piece of the car, it doesn't tell you how long it takes to get there. You forgot to look at the traffic, the weather, how much caffeine the driver drank. So when you look at one piece to the puzzle, it tells you a part of it and it can help. And there's certainly have drugs that have helped with individual genes, but we have to look at the whole system. If you walk in New York City now and draw blood on 100 people, 3 of them will have the chromosomal changes of leukemia, yet they won't have leukemia. So they need a host that's receptive in order for the leukemia to be a leukemia. And so we need to figure out a way also not just to measure the genes, but to measure the rest of us.
And let's make clear, we have not recommended that you drive to Denver.
So that relates to my question is, what do you think is the future of diagnostics then?
That's a good question. Who wants to take it?
I think in pathology, and we want to talk about this in regard to cancer, that there's a lot of potential now in molecular diagnostics using our understanding of the genome and how they relate to human disease, so that we'll have more precise diagnostics. Pathology is going to become a more important specialty, and it'll drive more precise therapy for the patient.
But there's new levels coming, right? There's more dimensions. You could take a drop of blood from a patient. In 1976, if you thought you were pregnant, we took a tube of that blood and we injected it into a rabbit. And 5 days later, we killed the rabbit. If the rabbit's ovaries were enlarged, you were pregnant. That was the state of the art. And then along came protein tests. And for one protein, we could say whether you were pregnant or not. Well, rabbits of the world rejoiced. And at the same time, we radically changed maternal and neonatal health. Well, we now have the ability through a drop of blood of looking at all the proteins in the body, the conversation that's happening in the body. And so technology is going to allow us to query that conversation and do the right thing in the right patient. And it ain't that far away.
We actually now have the technology to take a drop of blood from a pregnant woman and detect the minute amount of DNA from the fetus that crosses the placenta and gets back into the maternal blood, which has led to a new diagnostic for Down syndrome that's not
You don't have to
an
stick a
invasive
needle in it anymore and
diagnostic,
try to
right?
biopsy the chorionic villus.
Exactly,
Wild.
right. It's amazing.
The gentleman right behind you.
Would I be wrong in thinking that the traditional American notion about entrepreneurial and the government funding some of the very basic research that Carla talks about has in fact changed significantly, and that spirit is moving more towards the Singapores and the China, where the government really is funding not directed research towards a particular disease, but in general stuff, which is the foundation for a lot of the information yet to come. So in some sense, it's our Congress, which really needs to be educated to rethink where their funding priorities are.
I think one way of answering that is we get a lot of postdoctoral fellows from around the world, and I'm talking in general, medical schools that are doing biomedical research. And up until the last few years, almost all of those postdocs who came from China and Singapore, as well as Europe, they wanted to stay in the United States for their career because the money was available, it was flexible, they could be entrepreneurial. More and more now, they come and get their training here, and then they go back because of the money. Yeah, particularly China and Singapore.
Well, they also can't get visas to work here, but
James Fowles in our magazine is constantly bemoaning the visa problem and the drain of not being able to keep in foreigners who come and get great education here and should be incentivized to stay. Gentlemen?
I had a quick comment and a question. So first, I thought it was really interesting that Y Combinator is now investing in Biotech, which Y Combinator was an investor in Airbnb, I believe, originally. Also, I'm a graduate student at UCSF, and I saw how Susan Desmond-Hellmann was getting companies like Pfizer to invest something like $100 million in UCSF and its future. How do you see, these types of biotech companies investing in my future and the future of basic science?
I can take a crack at it. Years ago, the companies would give research institutes and academic medical centers money that was less directed. Like, we'll give you $50 million to do a lot of research, and we'll share in whatever IP occurs. Now, it's more directed around specific projects that, a company's interested in inflammatory bowel disease. They go to a university that's really strong in that. They develop a partnership. They put money into it. The university invests in that, and they work at it together going forward. So it's more targeted, which actually I think is better, because sometimes directed research with the right teams, gets you a product faster.
Well, here's the good news. The good news is there's still money from the big drug companies. And what's really cool is, as Dennis said, when they participate now, they can actually help move the research in a direction that is of interest to them and that would lead maybe to some kind of commercialization, whereas if you just left it in my lab, it could go in a random walk. So that's the good news. The bad news, I think, because we're doing this matchmaking with pharma too, including Pfizer. And the bad news is that, again, they'll come to us and we'll give them a little Chinese menu of research that they'll say they're interested in inflammation. We'll give them a Chinese menu of people at Stanford doing inflammation research. And then they'll always go for the one that's most de-risked. And they'll say, then we'll say, well, wouldn't you mind supporting this one? This could be, if it worked, it would be awesome. And they'll say, no, we'll stick with this one because it's less risky.
You should give them a menu of the ones you wanted them to fund.
Yeah, and you mean instead of any, because then they wouldn't fund any. They're really smart.
This trend, to me, this trend is so worrisome. Big pharma, again, is risk averse, and they're slow, and they're methodical. And when you have all your great ideas funded by big pharma, and they have the rights to intellectual property, the chance of it benefiting you or I in the short term is almost zero. The benefit of the 90s is we quickly go to a biotech company, go to a Y Combinator company, and then they're aggressive with this entrepreneurship, got it into patients right away, and proof of principle, and we got things happening and patients benefited. If we're going to go the way of relying on big pharma to fund our research in our institutions, we ain't going to be that innovative.
So, as we're about to end, we're going to end on some kind of optimistic note, for which reason I will ask, do any of you have any kind of discoveries that you've been working on that you're excited about, that you are able to talk about because you're not under gag order for various IP protection?
Well, I'll personalize it. My group made a discovery that ketamine is a rapidly acting antidepressant. Most antidepressant drugs take weeks, months to work, and they're not particularly effective. A decade ago, we found that it can work within a few hours. So at a minimum, it's a breakthrough that it's a new mechanism. And the most positive would be that it'll be developed. And so now J&J is developing ketamine in clinical trials.
We worked on a drug that bound to a receptor in breast cancer, and it stopped the receptor from hitting another receptor. And we worked on the drug for 10 years. A year and a half ago, it FDA approved. 4 months ago, it was shown that prior to surgery, so before you even operate, if you give this drug, 40% of the women who were considered, quote, inoperable, you can't even find tumor at the time of surgery. And a company called Genentech now has this on the market for the treatment of breast cancer.
So we can prevent the onset of Alzheimer's disease in mice. I know it's just mice, but by studying the developing brain and trying to understand why it is that kids can learn so much better than adults can learn, we found underlying molecular mechanisms and druggable targets that turned out to be really important in the adult brain and the way we encode memories. And we have in our brains a kind of brake on learning, and one of our molecules is one of these brakes. If you take away the brake, then in mice, they're smarter, and it also makes them resistant to the effects of all the bad beta amyloid stuff in Alzheimer's disease.
This is all great and exciting. I'm going to put in a completely random plug for a certain prestigious scientific institution in Boston where I teach science writing. So many of the things you've been mentioning, my kids have been talking about. So this is great because you are going to point the way to the future. We're going to have a new journal of failed results that will make this data available. I think it's an idea that's come up at Aspen before, and its time is ripe. But the time is really ripe for the kind of general unrestricted funding you're all asking for, and I hope we find ways of convincing the Congress to make it possible for all of you. Thanks for coming today.
Thank you.